Prokaryotes can thrive in the harshest environments found on Earth, from scorching volcanoes to salty waters to the frigid Antarctic. To survive in such extreme environments, most bacterial and archaeal cells are coated with a proteinaceous surface layer called the S layer. S layers are composed of repeating units of S layer proteins, which are the most abundant family of proteins on our planet due to their high copy numbers on microbial cells. This is Caulobacter crescentus, a freshwater gram-negative bacterium with a unique cell shape. Its S layer is a masterpiece of nature, comprised of planar tiling of RSAA protein units arranged in a hexagonal pattern. The outer domain of this S-layer protein forms a continuous lattice on the cell's surface, while the inner domain binds to the O-antigen sugar moieties of lipopolysaccharide, anchoring the S-layer to the cell. RSAA travels from the outer membrane to the end of the lipopolysaccharide molecules and connects into the existing S-layer on the cell's surface. Our second example takes us to the murky depths of the Dead Sea, where a mesophilic archaeon called Halophorax volcanii is found. The S-layer of this organism is crafted from the CSG protein, which is assembled into a hexagonal sheet with a markedly different arrangement to bacterial S-layers. Like many S-layer proteins, CSG has the ability to shapeshift and morph into different oligomeric states, such as pentamers and hexamers, that allows it to seamlessly cover the curved surfaces of these archaeal cells without any gaps. As illustrated by the two examples, S-layers are abundant in microorganisms across domains of life, from bacteria to archaea. Despite their abundance, they are far from perfectly understood. Future research is urgently needed to illuminate the secrets of these fascinating prokaryotic cellular molecules.